So let's talk about this very interesting dream challenge. So dream is a very famous challenge in systems biology. So it stands for dialogue on reverse engineering applications and methods and so on. And the dream challenge is there have been several rounds of the dream challenge, and there's typically a conference every year that where the results of these dream challenges are presented and so on. You can see the dream challenges in, so dream challenges uses crowdsourcing to solve complex problems. There are 60 plus challenges with 30,000 plus participants and 100 odd publications that have come out. So DREAM stands for Dialogue on Reverse Engineering Assessment and Methods. I said applications originally. It's a dialogue on reverse engineering, assessments and methods. And there are many challenges. So one of these past challenges is the disease module challenge, and here you go. This was a few years old, very interesting, where one had to systematically assess module identification methods on a panel of diverse genomic networks and discover network modules or pathways underlying complex diseases. So I'll tell you what the challenge was in a moment. So 6 different types of genomic networks, as you saw in the previous in the website, and there are 2 challenges, sub challenges that were there. So one trying to identify communities in individual networks, and the other trying to identify communities across all these networks, because the proteins are conserved or mappable from one network to the other, right. So the first one has 17,000 genes, a lot of them are shared, these nodes are shared with the second network, but it's a different set of interactions that you will see in the second network. The third network is much smaller, fewer nodes, but still mostly a subset of what you see in the previous networks. I don't remember what is the exact intersection in terms of nodes between all of these networks, but they are very much mappable. And we didn't have that information in the initial part of the challenge, but this information was then supplied later on. And how were these modules assessed? They were ranked by evaluation against 180 genome-wide association study data sets. They are called GWAS data sets, using a tool called Pascal, which does fairly advanced statistical analysis to determine if a given module is enriched or not for a given set of predicted genes in a module, predicted nodes, proteins, whatever. I use these terms interchangeably because you have these networks where every node stands for a gene or a protein and so on, and then you predict modules, right. You say that this particular subset of nodes is a module. Then is this disease relevant? Does it have enough genes that are associated with the disease and so on? What were the challenges? The first challenge was that we had anonymized networks, meaning we just had 1, 2, 3. I had no idea what protein this was. So I couldn't use any other biological information or any information from another database to predict if this protein is going to be involved in disease or not. And there were no ground truths available to train, meaning we didn't know what were the modules, what would be a correct module, what would be a wrong module. So which means that we had to only do unsupervised clustering. And we had very limited submissions and evaluations, and the evaluation itself was very time consuming. Not that it mattered, because we would anyway get the algorithm evaluated only once or twice, but then even when we had to do it locally later on, it would take a large amount of time simply because we had 180 data sets to compare against. And the modules themselves were of a very specific size that were required by the challenge, meaning you could give modules as small as 3 nodes, but no larger than 100 nodes. So this was a central restriction to this challenge, and this was clearly a challenge for the classic community detection algorithms, because the classic clustering / community detection algorithms would predict modules of arbitrary size. So there was no way to obviously tune them so that they would have modules sizes in this prescribed range. And we also had to identify non-overlapping modules we tried to show later on that this was a bad idea, simply because in biology it's very difficult to bucket one particular gene or a protein into one particular module alone. There's so much of cross functionality, cross interactions and so on, cross talk, that one particular node typically interacts with so many other nodes in different modules. Right? This is not for all nodes, but there are genuinely a few nodes that are persistent and present across several different modules in the network. The big question for us is, how do we extend classic community detection algorithms to work on biological networks? And this is typically a very fundamental question in bioinformatics. You have an algorithm from computer science, you have various excellent algorithms that exist. Like it could be a local, it could be an edit distance, local alignment kind of algorithm. How do you adapt it to take into account the challenges that you encounter in biology? It could be in terms of uncertainty, it could be in terms of various other unique biological problems that you have to deal with. The challenge summary is as follows. There were 42 single network and 33 multi-network methods that were contributed by a host of teams and participants who were a part of this challenge. And it's a very nice consortium paper that appeared in Nature Methods, which gives you a pretty good rundown of a lot of these methods, and the supplementary material includes many other methods that were actually proposed and so on. We published our own paper on adapting community detection algorithms for disease module identification in various biological networks as a separate paper focusing on our methodology. We were one of the top methods in the challenge, although we did not win it, and our method had some unique contributions which I am going to talk about. And it gives you a perspective of how do you take an existing algorithm and start modifying it and molding it to work on biological data sets. And these are some of my collaborators on this project. There's Bitika Tripathi, who was my MS student, and Karthik Argasan again, who was an MS student, jointly with Professor Ravindran. And we also had collaborators from our department and from abroad. So we took three different approaches, trying to extend classic approaches, trying to identify core modules, and most importantly trying to identify overlapping modules, which means that a particular node, a particular protein could be assigned to more than one module in the network. So first let's talk about some of the classic algorithms which give us some sort of baselines. So we first need measures of community quality. What is a good community? Right? So there are measures such as modularity. I again would like to remind you that we discussed modularity briefly a few lectures ago, which essentially, this was a definition given by Newman. Do you have a higher than chance edges within a group compared to a null model, right? So do you have too many edges, a very tightly knit group compared to a null model where you would have randomly thrown edges all over the network. And then there is this measure of conductance, which tries to measure how isolated nodes are in a network. And then there is this algorithm called SEAL, which tries to take middle ground between modularity and conductance. So modularity tries to measure internal density, whereas conductance tries to measure isolation and so on. And then there is Markov chain clustering, which is a random walk based approach. This was also something that we used. We then went ahead to identify core modules using multiple approaches. The idea was to identify small and structurally well defined communities, and these are more compact than what a normal community detection algorithm gives, right. So the idea is, our hope was that this would be more disease relevant. And we also performed ensemble clustering based on consensus from a number of algorithms. So not from a single community detection algorithm, but from multiple community detection algorithms, trying to build a consensus module which tries to assign modules that are proteins that are predicted to be in the same module across algorithms. We try to put them into the same module. And then we also tried to do perturbation to identify persistent communities. This was a very interesting approach. I will talk about this in greater detail. And trying to identify multiple core modules by taking a large module and breaking it down based on various metrics. So first thing is ensemble clustering. So how do we take consensus from other approaches? Let's say, for example, we here used Markov chain clustering, modularity maximization, and this method called SEAL to cluster the network. What did we do? We took these different algorithms and applied it on the same network multiple times. We took different algorithms and applied it on a given network. So node N1 falls into community 1 according to algorithm 1, community A according to algorithm 2, and community X according to algorithm 3. But if you see N3, it occurs in community 1 according to algorithm 1, community 2 according to community A according to algorithm 2, and community Z according to algorithm 3. So if you see, these three algorithms give three different clusterings or communities. So in the first one, N1 and N3 are in the same community. In the second one, all three are in the same community, and in the third one, all three are in different communities. But now I can compute the similarity between these different clusterings. So here, what is the similarity between the memberships of N1 and N2, right? So 2 out of 3 times it has the same membership, right? So that is captured here, and this number is put into this square, and obviously into this entry also of the matrix. And obviously the diagonal is going to be the same because it is the similarity of an algorithm of a vector with itself, right? So it's 1ax versus 1ax. So here this is going to be 1ax versus 2ay, and that's going to have a similarity of just 33%. Right, so for either N1 versus N2 or N2 versus N1, that's going to be the similarity. And similarly for 2ay and 1az, you again have a similarity of 1 by 3. We then use Jacquard similarity between these vectors to build a similarity network. So whenever the similarity is greater than 0.5, these numbers are greater than 0.5, we use them to build a network, right? So whatever is greater than 0.5, we keep them, less than 0.5, discard them. So you now have a different network, and we ran modularity maximization on this network. What is modularity maximization? It's a discrete optimization problem, maximizing Newman's measure for modularity that tries to capture how well connected a given set of nodes is compared to what you would expect by chance. Okay. And this is something that's very interesting and a very biologically motivated methodology. So can you identify persistent communities based on perturbing the network? So what did we do? We randomly dropped 1% of edges in the network about 100 times. So we took this network, threw away 1% of the edges, some random 1% of the edges. So you get a network. Repeat, you get another network. Repeat, you get another network. You repeat it 100 times, you get 100 different networks that have 99% of the edges that were present in the original network. So all these networks are slightly different. Obviously 99% of the edges are the same, but they are still slightly different. Now let's identify communities in these resulting networks. Now you perform community detection on these networks, and you have 100 different community assignments compared to a few different algorithms we talked about in the previous slide. Again, use Jacquard similarity between these differing community assignments. You would imagine that they will differ slightly simply because of the changes in connectivity from the original network to the perturbed network, and again perform modularity maximization on the resulting network. And that was one of the approaches. The next approach is trying to prune large networks to improve the quality. So you try to find core communities which have minimum outgoing edges and so on. So essentially we want to try to prune out less important edges from a module. So, or basically pick nodes that form much better communities. How do we do this? So good communities should have a lot more internal edges, intra-edges than inter-edges, meaning internal versus outgoing edges. So we give a core score that tries to capture what is the fraction of outgoing edges from a vertex by the number of internal edges in a vertex. And we use this to improve our community predictions. So we took large modules and downsized them to prune to the top 60 nodes based on the core score, and this is because we realized by looking at literature and so on that the average size of a disease module is only about 60. So we didn't even go as far as 100 that was permitted by the challenge. The challenge said anywhere between 3 to 100 nodes are acceptable in a module, but we basically said we will go with modules of size at most 60. And here is a rigorous comparison of all the methodologies. So, all the columns that you see here, save for the last column, are from our predictions, and the last column is a method called diffusion state distance algorithm, which was declared the winner of the challenge based on the fact that they were predicting a large number of modules. But if you see, the ratio of the predictions may not be the best for their algorithm. For example, there are cases where we've been able to predict as many as 22 modules with only 462 modules even predicted. So, they predicted 1020 modules, out of which 24 were enriched, and so on. Right. Of course, in the second case, they predict 445 modules, and they predict 19 enriched modules from 445. But if you see, we typically have a much better hit ratio. Right. Our numbers are typically smaller in terms of the number of predicted modules in most cases, except in this case. And here you see the hit ratio, right. So, the hit ratio for us is high in basically all the cases, although it is not a single algorithm that is the highest in all cases. So, to summarize, this was a very interesting challenge where we had to work on multiple biological networks and make predictions of what would be the most plausible communities just based on the network structure. Remember, we had no other information. I didn't know what protein it was, what is its biology, or any of those things. I knew that there was a protein A which connected to proteins B, C and D in network 1, but the same protein A may have connected to I have a protein A that connected to nodes B, C and D in network 1, but the same protein may have connected to B, C and E in network 2 and so on. I had this 1, 2, 3, 4 or ABCD as I showed you in an early slide as the network, and just based on this network topology, I had to make predictions on what would be the most optimal communities and so on. And there are many classic community detection algorithms, we already studied a few, but we just couldn't take them and directly dump them on biological data sets. We had to tweak the algorithms, we tried to break down the modules, we tried to identify overlapping modules, we tried to identify persistent modules by basically perturbing the network and so on. So, we used a number of different techniques to adapt existing community detection algorithms to work on biological data sets, biological networks. So, this was one application, and we will look at a number of applications in the rest of the classes. See you.